Hello everyone, welcome back to Mahi's Life Science channel. In this video, we will discuss about the principle, material record, procedure and interpretation of Gram staining technique. Gram staining is a laboratory technique used to differentiate bacterial species into two major groups based on their characteristics of their cell wall. It was developed by the Danish bacteriologist Hans Christian Gram in 1884. The process involved applying a series of stain to bacterial sample which result in either gram-positive or gram-negative organism based on their color and the structure of their cell wall. The distinction is very important because it helps in identifying bacterial species and determining appropriate treatment options. Principle of Gram staining. The principle of Gram staining is based on the differences in the structure of bacterial cell wall. The staining process takes advantage of how bacterial cell wall interact with various chemical stains and reagent, leading to a color differentiation under microscope. The key differences lies in the thickness of the peptidoglycan layer in the bacterial cell wall and the presence or absence of an outer membrane. The structural variation influences the ability of bacteria to retain primary dye crystal violet after a decolorization step, usually with alcohol or acetone. Gram-positive bacteria have a thick peptidoglycan layer and that is the primary component of their cell wall, but they lack an outer membrane. The thick peptidoglycan layer traps the crystal violet iodine complex inside the cell that we call as an CVI complex. This complex is not washed away during the decolorization step. So the bacteria retain their purple color from the crystal violet stain. Gram-negative bacteria have a thin peptidoglycan layer and a more complex outer membrane that lies outside the peptidoglycan layer. When the decolorizer, either alcohol or acetone is added, it dissolves the outer membrane and causes the thin peptidoglycan layer to become permeable, allowing the crystal violet iodine complex to be washed out of the cell. The bacteria are then stained by the counter stain safranin, which give them a pink or red color. Materials record: microscopic slide, bacterial smear, crystal violet, primary stain, iodine solution, mordant, decolorizer, usually alcohol or acetone, safranin, counter stain, distilled water, staining rack, paper towel or blotting paper, Bunsen burner for heat fixation. Procedure: prepare the bacterial smear. Transfer a small amount of the culture to the slide using a sterile loop and mix it with a drop of water on a microscopic slide. Heat fixation: pass the dried slide through the Bunsen burner flame several times to fix the bacteria to the slide. Staining with primary stain, that is crystal violet. Place the slide on a staining rack and flood the smear with crystal violet and allow it for 1 minute. Rinse with water. After 1 minute, gently rinse the slide with distilled water to remove the excess crystal violet. Iodine treatment, that is mordant. Flood the slide with iodine solution and allow it for 1 minute. Then rinse with water. Rinse the slide gently with distilled water again to remove excess iodine. Decolorization. Add the decolorizer, which may be either alcohol or acetone, to the slide for 5 to 10 seconds or until the run of the liquid is clear. Rinse with water. Immediately rinse the slide with distilled water to stop the decolorization process. Then counter staining with safranin. Flood the slide with safranin for 1 minute. Rinse and dry. Rinse the slide gently with distilled water to remove the excess safranin. Examine under the microscope. Place the slide on the microscopic stage and observe it under oil immersion, that is 1000x magnification. Result and interpretation. Gram-positive bacteria. These bacteria retain the crystal violet stain and appear purple under microscope due to their thick peptidoglycan layer in their cell wall. Gram-negative bacteria. These bacteria lose the crystal violet stain after decolorization and take up the safranin counter stain, appearing pink or red under microscope. The cell wall have a thin peptidoglycan layer surrounded by an outer membrane. Example for gram-positive bacteria, they are Staphylococcus aureus, Streptococcus pneumoniae and Bacillus anthracis. Example of gram-negative bacteria, Escherichia coli, Salmonella enterica and Pseudomonas aeruginosa. Thank you. Like, share and subscribe for more videos.